Hello everyone, my name is Grace and I'm excited to share the results from our paper, The Comparable Tumour Microenvironment in Sporadic and NF2-schwannomatosis Vestibular schwannoma, published in Brain Communications. In this paper, we investigate the tumour microenvironment of vestibular schwannoma, which are brain tumours that develop from the Schwann cells that myelinate the 8th cranial nerve. These tumours can compress the brainstem and damage the inner ear, resulting in sensory neural hearing loss, imbalance and tinnitus, which can lead to a poor quality of life. Vestibular schwannoma can arise either unilaterally on one side in sporadic non-inherited cases, or bilaterally on both sides in the rare tumour predisposition syndrome NF2-related schwannomatosis. In NF2, vestibular schwannoma are also accompanied by other tumours such as spinal and cranial meningioma, ependymoma and peripheral schwannoma. We wish to compare the vestibular schwannoma from these two patient cohorts with the hypotheses that similarities in tumours from sporadic and NF2 patients could allow their co-recruitment to the same clinical trials. Also, treatments that are effective in one population may be applicable to both populations. And finally, preclinical research findings in one group may be applicable to the other group, thus widening the pool of research resources available. Hence, the aims of our study were to evaluate the tumour microenvironment of sporadic and NF2 tumours by comparing their gene expression, signalling pathways, cell abundance, and immune niches. To evaluate these aspects, we used a previously published single-cell RNA-seq dataset from 3 sporadic vestibular schwannoma, as well as 3 published Affymetrix bulk RNA datasets for gene expression and predicted cell type abundance. We validated this with qPCR for gene expression and Hyperion imaging mass cytometry. Using one of these 3 datasets as an example here, we identified many differentially expressed genes between the tumours and nerve samples. These significant genes were enriched in many of the same pathways in both NF2 and sporadic vestibular schwannoma, shown in this dataset with an overlap of 69%. By directly comparing the gene expression of the sporadic and NF2 tumours, we found that principal component analysis also revealed a strong overlap in these tumours, and that although there were significant differentially expressed genes present, none were consistent across the 3 datasets. As such, we showed strong similarities between sporadic and NF2 tumours in their gene expression and their enriched signalling pathways. Next, to assess cell abundance in sporadic and NF2 tumours, we used the published single-cell data and 3 bulk RNA datasets to predict cell type abundance in these tumours. The single-cell data revealed that these tumours were variable in their relative abundance of components, but on average, the sporadic tumours were composed of 30% Schwann cells and 51% macrophages. The single-cell RNA-seq data was used to generate a list of genes pertaining to each cell type that was then used to deconvolve the bulk RNA datasets. We did this using CyberSortX, a deconvolution algorithm, and we have published this vestibular schwannoma-specific signature matrix for open access use. This revealed no significant differences in predicted abundance of cell types between the 2 tumour groups, with both displaying high levels of macrophage infiltration. Furthermore, there were no significant differences in macrophage gene expression between sporadic and NF2 tumours when assessed by qPCR. From these published datasets, we went on to assess cell type abundance and to identify immune niches using high-dimensional imaging. Tissue stained with metal-conjugated antibodies can be imaged to identify tumour microenvironment components and niches within the original tissue architecture. Using imaging mass cytometry, we noted immune-rich niches of T cells and macrophages in both sporadic and NF2 tumours, especially in highly vascular areas. When segmented into single-cell data, the relative cell type abundances were found not to be significantly different between sporadic and NF2 tumours, as they were composed of an average of 49% Schwann cells and 31 to 36% macrophages. Together, the data in our study identified strong similarities in the gene expression, signalling pathways, cell abundance and immune niches of sporadic and NF2-schwannomatosis vestibular schwannoma, which reinforced the hypotheses that these patient groups may benefit from the same drug treatments, co-recruitment to the same clinical trials, and that preclinical research using samples from one group may be applicable to the other. And with that, I would like to thank all co-authors, our acknowledged colleagues, and the funding bodies that supported this study, as well as yourself for taking interest in our study through this video abstract.